Welcome back, everyone. Today's lecture is going to focus on Chapter 8. It's entitled DNA Replication, Binary Vision, and Mitosis. This is a really interesting chapter because we're going to be exploring DNA a little bit more. We're going to take a look at exactly what needs to be done for DNA strands to replicate themselves. And we're also going to start talking about mitosis and meiosis. These are two forms of cell reproduction that our body utilizes. And throughout chapter 8, we're going to focus mostly on mitosis. And then in our following chapter, chapter 9, we're going to take a look at meiosis in a more detailed scale. But for now, what I want to do is I want to remind you that one of the characteristics of being a living entity is that there should be a sign of reproduction. And this reproduction can either be done asexually or sexually. So this should remind you of our conversation that we had in chapter 1. As you might recall, asexual reproduction, A means without sex, means that you just have one organism that's able to duplicate itself and create a next generation that is identical because there's only one source of DNA. So for instance, you have a picture right here of some bacteria, or I should say a bacteria cell. And what it's showing you is that one bacteria cell will go ahead and duplicate its DNA, and then it will physically split the cell in half, creating two what they call daughter cells. These are going to be two new generation cells that are identical to the original cell because they only had one source of DNA. This should also be the part where I put my little disclaimer, which is that the only way you can get variety in an asexual reproduction is if there is a mutation, a random change in the DNA that appears. Beyond that, everything will be identical from the original source. On the other hand, if you do sexual reproduction, you're going to get more of a genetic variety. And part of that is because you involve two organisms, two sources of DNA, so that the offspring becomes a hybrid or a mix of those. In order for the body to do sexual reproduction, we're going to have to use this form of cell reproduction called meiosis. Meiosis is a way for us to produce cells that only have half our genetic information. Now, we as humans have 46 chromosomes, or we can say we have 23 pairs. And the reason we can say 23 pairs is because we get half our DNA from mom and the other half from dad. Because we get two pairs of everything, we like to say that we are diploid. DI meaning two of everything. When it's time for us to produce our sex cells or our gametes, so our sperm and our egg cells, we are not able to take all pairs of DNA and pass it on. Instead, we can only take half of our pairs. So instead of creating a diploid cell, we're going to create what's called a haploid cell. A haploid cell only has half the DNA or 23 chromosomes inside of it. So meiosis in the human body is used to produce our reproductive cells, our gametes, so our eggs and our sperm. The rest of our body cells, like our skin cells, our liver cells, our kidney cells, those need to have 46 chromosomes at all times. So when those cells need to reproduce, they can't use meiosis. Instead, they're going to use mitosis. Mitosis will allow us to produce daughter cells or the next generation of cells that are identical to the original cell. So we start off with 46 chromosomes, we produce 46 chromosomes in the next generation. This will take place in what we call somatic cells. Somatic cells are all your cells in your body except for the reproductive cells. So your hair cells, your skin cells, your stomach cells, your intestines, anything in your body that has nothing to do with sperm and egg will always do mitosis. When the cells replicate, they produce cells that are identical to what they started with. We started with 46 chromosomes. We're going to end with 46 chromosomes. Meiosis will exclusively be used for our gametes or our sex cells. So if it's time to produce sperm, if it's time to produce eggs, then we'll use meiosis because that will generate haploid cells, cells that only contain half our genetic information. So for instance, we can take a look right here at a little life cycle of humans, and we can see that in our adulthood, or I should say once puberty is reached, we're able to utilize meiosis to produce our gametes, our sex cells. So here's the sperm, here's the egg. Each of them have to use meiosis to be produced because they can only carry 23 chromosomes. When it's then time for fertilization to occur, meaning that the sperm enters into the egg, we then see that the 23 chromosomes from mom align with the 23 chromosomes from dad, and now you have a zygote or a fertilized egg that has 46 chromosomes. Beautiful. So that's the number that we're looking for. Now, to go from a one-celled organism to a multi-celled organism, like this little baby right here, what we're going to have to do is mitosis, produce cells that are identical in number, so 46 to 46, so that we can start growing our different tissues, our different organs that all contain our 46. And in fact, even today, as we are maybe perhaps done with growing, we are still maintaining our body. And in order to do that, mitosis plays a huge role in that because it will be able to allow us to replicate our somatic cells, our everyday cells, with an equal amount of genetic information as we go from one cell to their daughter cell or to their next generation cell. So keep in mind, mitosis is for your somatic cells, and your meiosis is going to be for your gametes, which are going to be your sex cells. So the only time you use meiosis is if you're making sperm or egg. Anytime else, it will be mitosis. And we can even take a look at this diagram right here that shows us how we explain going from a zygote or a fertilized egg to a multicellular organism. So here you can see your first 56 days of development. Starting with day one, we see a zygote, a fertilized egg, and that one egg will turn into 2 cells, will turn into 4 cells, into 8, into 16, into 32, until it literally is just a ball of cells that will continue to divide and forming what we call our germ layers. The germ layers, you can see there's different colors representing them here in the yellow, the orange, and the blue. The germ layers will have specific destinations. Some of them will become your nervous system, some of them will become your bones, some of it will become your muscles. So as the development continues, you see that a ball of cells starts to take shape. Look at this beautiful little fetus continue to grow. And as you can see, our little life cycle on this picture ends at 56 days. And as you can see, you now have a complex multicellular organism, all due to mitosis, continuously producing cells that are identical in content in 46 chromosomes as the other cells. And just a little fun fact, when you look at your zygote, your fertilized egg right here, the first picture, you see how you have like this little halo around it? It turns out that the minute that the sperm cell enters into the egg, and you're back at 46 chromosomes, this little halo will form. And this prevents any additional sperm cells from entering because humans need 46 chromosomes. And any deviation from that, so it's like a second sperm were to enter or a third sperm cell, would prevent the zygote from successfully doing mitosis, and it would cause it to stop replicating. So that's a little fail-safe that the body does to make sure that 23 plus 23 equals 46. The other thing that I want to mention is, mitosis will help us replace and grow cells that always have 46 chromosomes, but there might come a time where we no longer need certain cells and we would want to get rid of those cells. So cell death is a part of our little discussion right here. And one of the ways that cell death can occur is through what they call apoptosis. Apoptosis is known as programmed cell death, and often scientists will refer to it as cell suicide. The reason it gets that name is because the cell will receive a signal, whether it's internal or external, and it will literally pack up all its organelles, it will destroy its DNA, and it will implode, so it will fold into smaller compartments. And eventually it gets into small little pieces that a white blood cell, like a macrophage, will come around and it will phagocytose it. It will bulk eat it and get rid of it. It is a very controlled method of getting rid of the cell. It doesn't cause any inflammation or side effects, so it is a preferred type of cell death. In case you were wondering, if the apoptosis is the programmed cell death, then the other version of cell death is called necrosis. Necrosis is very traumatic cell death. In this case, the cell will often swell and literally explode, causing inflammation to occur all around it. But that's a conversation for another day. For now, I want to show you the picture of our little frog right here. So frogs do external fertilization, which means that they release their egg and their sperm into the aquatic environment after they have sex. The egg and the sperm are chemically attracted to each other, and once the egg is fertilized, it will use mitosis to produce tadpoles, which you can see swimming around water sources, especially for me around here in Florida. Come summer months, all the lakes and even some of the pools can have tadpoles just swimming inside of them. The tadpole will continue to grow through mitosis, and at a certain point, it will metamorphosize so that it can move from an aquatic environment to land. And when it goes on to land, it no longer needs the tail that the tadpole has because it's not swimming anymore. The way that you can describe the tail disappearing would be through apoptosis, programmed cell death. We also see some examples of apoptosis when we take a look at embryology or how embryos are able to develop. And for instance, if we were to take a look at our chick and our duck right over here, very early on in their development, when their digits or their feet develop, you can see that there's webbing in between the toes. For the chick, that webbing will disappear by the time it's born. For the duck, that webbing will stay behind. And the reason for that is because the cells for the chick went ahead and did apoptosis, so they died off, hence the webbing disappeared. And for the chick, those cells never received the signal for apoptosis, so they continue to thrive, hence the webbed feet. It also has to do, of course, with the fact that ducks tend to be more of an aquatic species versus the chick. And don't feel left out, even in humans, early on in our development, we also have webbing in between our fingers. And right before birth, apoptosis will occur to those cells and the webbing will disappear. Programmed cell death will occur and you'll have your free fingers or your free digits by the time birth occurs. Now let's go back to DNA. So if you want your cell to replicate, I think it goes without saying that we also need to have a way to replicate our DNA. Because remember, we want to have our cells, when we do mitosis, consistently have 46 chromosomes. So that means that there has to be a way for us to double up our original number as we get ready for our cell to go from one cell to two cells. So we need to talk about DNA replication. How do we double our DNA? And it turns out the way that's going to happen is through a semi-conservative model. And semi-conservative means that we're going to use 1/2 of the old piece of DNA to generate 1/2 of the new DNA. So you'll always have one part that's old and one part that's new. To put it very plainly, if you think of your DNA like your little ladder, your little helix, what we're going to do is we're going to split the helix open, and then we're going to use the old part of the helix as our base so that we can build a new strand that's complementary. Remember, A goes with T and G goes with C. The end result is that you then have two new pieces of DNA, but notice how each piece is part old in red and each piece is part new in blue. So that would be an example of semi-conservative replication, one old and one new strand. In order for this to occur, there's going to be a variety of enzymes that we're going to have to utilize. So I definitely want to talk to you about those. But before we do that, let's take a look at figure 8.4. This one shows us a much better representation of the DNA replication than the one I just drew. So as you can see on top right here, you have your double-stranded DNA. And as we can see, it's going to start opening up. And then using the old piece as a template, we can see that here in the light blue, almost silver backbone, you can see a new strand of DNA being built that's complementary to the old strand. So you can see the old strand has a T, we're going to match that with an A. The old strand had a Z, we're going to match that with a G. So your end result is that you go from one strand to two strands And for those two strands, you have one part old and one part new. They like to say it's one parental, because parental is the old strand, and one daughter strand. Daughters are always used as reference to indicate the next generation. So it's semi-conservative, half old, and half new. Alrighty, so let's talk about the enzymes that are going to be needed to get all this done. You can see them listed right here on the slide. We're going to need the helicase. We're going to need the binding protein. We're going to need the DNA polymerase. That's the one that's going to do all the work. We're going to need a primase, and we're going to need a ligase. Now, on the next slide, the functions have been spelled out for you. So what I want to do is I want to go over to the next slide. I want to talk a little bit more, but please know that this slide right here has the list of all the ones that we're going to be discussing. So at the end of our lecture, you want to make sure that you know what the helicase does, what the binding proteins do, what the DNA polymerase does, and how that relates to Okazaki fragments. Where does the primase fit in, and where does the ligase fit in? So let's go over to the next slide and let's take a look at our illustration. So quick reminder, the replication of the DNA is going to take place in the nucleus of the cell because that is where you house your DNA. And what we're looking at here are going to be the proteins needed. So first up is going to be your helicase. And as you can see, the helicase is going to allow us to unwind the DNA. So just like a zipper, we're going to be opening it up so that we can get access to the little nucleotides that are in each side of the strand. Now, DNA has a tendency to coil back up. So what we're going to do is we're going to use our binding proteins to keep the strands open. So the helicase unzips, and then the binding proteins will keep it open. The next thing we're going to need is we're going to need to build a dock. And that is going to be done by the primase enzyme. The primase enzyme is going to build a short little RNA sequence that's going to be a primer. The reason we need this primer is because our next enzyme, the one that's going to be doing all the work, needs an anchoring space. It needs to dock, it needs to hold on to something before it can get started. And that enzyme is called the DNA polymerase. The DNA polymerase is going to help us make a complementary strand of DNA. So this is the enzyme that's going to read the old strand and produce the new strand. So it's going to read the A and match it with a T. It's going to read the G and match it with a C. And in order to get started, it needs a little primer. Now, I know the primer says it's RNA, but just so you know, the primer is eventually replaced with DNA. Now, here's the other thing about the DNA polymerase. It can only move in one direction. So as the DNA polymerase is moving, we see that with our two opposite strands, in one occasion, the DNA polymerase will move forward, and in another example, it will move backwards as it's reading it. When it moves it forward, it does what we call a leading strand. And I will show you this on an illustration coming up. The leading strand will be a continuous long piece of DNA. The lagging strand is when the DNA polymerase has to go in a backwards format. It's called lagging because the DNA polymerase can only make fragments when it does it backwards, not a long piece. These fragments are called Okazaki fragments. And in order to seal the fragments so that we get a long piece of DNA, that is then where we're going to be using our ligase enzyme. So DNA polymerase moves forward, we call that leading. If it moves backwards, we call it lagging. And lagging will give us Okazaki fragments, which need to be sealed by the ligase enzyme. Here at the bottom of the picture, you can see the use of the helicase enzyme. To separate, we can see our binding proteins that are keeping it open, and then we can see that our primer is actively building our little docking station for our DNA polymerase to take hold of. As the DNA polymerase gets positions, and you can see it right over here, looks like a little kidney bean shape, you can see that it's going to start moving, and as it's moving, it is reading the old strand of DNA, and it is matching it with a new piece. So this highlights the fact that it is a semi-conservative model that it's working on. Now, remember, the DNA polymerase are going to be moving in two different directions. If it's moving in a forward strand, so this would be from the 5 to the 3 prime, then what we see happening is that we call it a leading strand because it continues to make a long strand of DNA. So leading strand means the DNA polymerase moves forward and you have one long piece of DNA. If the DNA polymerase has to move the opposite from 5 to 3, then what we see happening is that it's going to create fragments. These are called Okazaki fragments, and this side is called the lagging strand. So lagging strand means moving backwards and creating fragments. Now don't worry, because the fragments, we're going to eventually have to seal them so that we can create a long piece, and that is where your ligase enzyme is going to come in. So the ligase enzyme is only active in the lagging strand because that's the only strand where you have little fragments that need to be sealed off. So here you can see it sees discontinuous synthesis produced by the Okazaki fragments. This is only when the DNA polymerase is moving in a backwards way, creating the fragments. All right, the distinction between leading, forward, and lagging backward strand. And last but not least, highlighting the fact that the ligase enzyme is only active in the lagging strand. It takes about 8 to 10 hours for our chromosomes to fully be able to replicate themselves, so to go from 46 to 92. Now, this is the part where I want to take a quick little break from DNA replication, and I want to mention this term called binary vision. Binary vision is seen in prokaryotic cells like bacteria and archaea. It is basically a form of asexual reproduction where the bacteria cell will double its DNA. And by the way, bacteria only have one chromosome, so they will double their chromosome and then they will split themselves in half. This is a form, as I mentioned before, of asexual reproduction. Eukaryotic cells can't do this because we have more chromosomes that are larger and more complex. And in order to get a view of it, you can take a look at the picture that's below, and it's showing you that the eukaryotic cell, we can have the nucleus fully intact, and inside that nucleus, all your chromosomes are being housed. You really can't see them until the cell gets ready for replication, whether it's doing mitosis or meiosis. If it gets ready for replication, it has to double its DNA, so it will go through the process of going from 46 to 92. And as it proceeds to start dividing and producing its daughter cells, we're going to notice that the chromosomes become more visible. And that's why in the second picture right here, you can see instead of the circle for the nucleus, you can start seeing these strings appearing. Those are the chromosomes that are becoming more visible. Here is a look at that binary vision that we were just chit-chatting about. So you can see it's very simplistic. This is done for prokaryotic cells like our bacteria and our archaea. You can see you have one strand, one circular chromosome. The DNA is replicated, and then the cell is physically split into two daughter cells. Each of them are identical because once again, there's only one source of DNA. Our eukaryotic cells cannot do this because we have a massive amount of DNA. So instead, what we need to talk about next is how exactly do we condense or do we store our DNA? And in order to do that, we have to talk about this thing called chromatin. Chromatin is the key term that we use whenever we take a look at condensed version of our DNA and our proteins that are associated with the DNA. And part of what I want to show you on the next slide is how we can take our long strand of what I call naked DNA, DNA by itself, and we start coiling it on top of each other. And as we're coiling it, we're going to create these things called nucleosomes. And nucleosomes utilize histones or proteins to wrap themselves and condense themselves down. And when we stack these nucleosomes together, we start creating chromatin. And it will keep condensing down until it starts to take a shape of a chromosome. Just to give you an idea of how much DNA we have in one cell, over 6.5 feet of DNA in just a single cell. And our body is made up of millions of cells. Each of our 46 chromosomes, if we were to sit there and like read out how many A's, how many T's, how many G's, and how many C's they have, it would take us over 2 million pages of typing it out. So we're talking about a massive amount of information. So the formation of the chromatin is really important because it allows us to take this really long strand of DNA and condense it down so that we can safely store it within the nucleus of the cell. Now, I do want to point out as we look at some of the images, you're going to see the mention of chromatin, T-I-N, and chromatids, T-I-D-S. And there's a difference in those key terms. Chromatin is when we're looking at DNA that has been condensed down and that is freely accessible for your body to use for things like transcription and translation. This is when the cell is just doing its day-to-day activity and is not focused on dividing. When it does become time to divide, what we see happening is that the DNA will make a duplicate copy of itself. So here's one, here's the original one. This, for instance, can be the duplicate copy. When that copy is made, it will hold on to it through the use of a centromere. And then what we have are two pieces of DNA that are the same. So what we're going to do is we're going to call them sister chromatids. So when you see the term chromatids, T-I-D-S, I want you to think about the fact that you have two pieces of duplicated DNA that are being held together. So when the cell goes from 46 to 92, it goes from chromatin to chromatids, because chromatin is just coiled up DNA, and chromatids means that you have double the DNA that you started with. So here on this illustration, for instance, you could see you have your naked DNA. This is what we're used to seeing when we look at our DNA models, but we have a massive amount of this. So what your cells will do is it will take the piece and it will use proteins called histones to coil it around. And these little coils are called nucleosomes. So whenever you see the term nucleosome, I want you to think, oh, that is DNA wrapped around a histone. And when you stack the nucleosomes together, it kind of looks like an old school telephone cord. You then call it chromatin. So as you can see, chromatin is just nothing but just condensed DNA that we're just pulling, making smaller and smaller so it can easily store. If that chromatin piece is then duplicated or replicated, we call them chromatids. So chromatin is just stored, condensed DNA, and chromatids means that you have two pieces, two sisters, two replicated parts. And this all will play a role into our cell cycle, because the cell cycle are going to discuss the different events or stages that all cells go through from their day-to-day activity up until the point that it's time for them to do a cell division. So for them to create their daughter cells, their next generation. Now, quick reminder, when it comes to cell division, you can have two options. You can have your meiosis or your mitosis. This chapter is going to focus on mitosis, and then in chapter 9, we'll take a look at meiosis. What you're looking at on the screen right here on figure 8.8 is an illustration of the cell cycle. As you can see, it's presented in a circular format because as one generation produces the next one will go right into the circle and continue on the process. Notice how the majority of the circle is represented by orange and it's labeled interphase. And that's because this is the day-to-day side of the cell. And then in blue, we have our cell division. And in our case, we're going to be looking at mitosis. So what I'd like to do now is talk to you about what happens into each of these phases of the cell cycle. So let's start off with the longest phase, which is interphase. Interphase is when the cell is doing its basic functions. So it's doing transcription, translation, it's doing its day-to-day stuff. So the kidney cell is filtering, the skin cell is protecting, the stomach cell is producing acid to help you hydrolyze your food. So the cell is just doing its basic functions. Interphase takes up about 75% of the cell's life cycle, and it's subdivided into three divisions. You have G1, which stands for gap 1, or some books will like to say growth phase 1. You have S for synthesis, and then you have G2, gap phase 2, growth phase 2. G1 is when you have a brand new cell and the cell will start growing in size and it will start being able to respond to the signals and starting to complete their basic function. At a certain point, the cell will then go ahead and duplicate its DNA. It will go from 46 to 92, and that will be the synthesis phase. This is when you go from chromatin to chromatids. As the DNA is duplicated, then we go into G2, which is the cell making sure that it has all the DNA and the proteins required to complete its cell division. Not all cells will actively divide. Some cells will stay in what we call G0. G0 means that the cell is functioning, but it has no intentions of replicating. Our neurons, our brain cells, are a good example of cells that stay in G0. We have no ability to replicate those neurons now. So what you're born with is what you're going to have for the rest of your life. This can be a temporary state in some cells. Some cells will go into G0. We see this especially in plants when the environment is unfavorable and they don't have enough food or resources to replicate. So they'll go almost in a hibernation state called G0, and then they can reverse it when conditions get more favorable. Once the cell is done with interphase, it's going to start looking into its cell division, into mitosis. Now, when we do mitosis, part of what we have to do is that the cell has to physically divide its DNA, because remember, it has twice as much DNA as it needs because it did the synthesis step. So in our cell division, we're going to have to divide that DNA back from 92 into two batches of 46. In order to do this, you're going to need some type of mechanism to move everything around. And that is where the mitotic spindle is going to come in. The mitotic spindle is a protein complex that will allow the movement of the chromosomes, and it will be able to organize the chromosomes. You can see them right over here, the little sister chromatids held together by the centromere. The mitotic spindle will be the one moving them around and separating according to what the cell needs to get done in its cell division. In our case, our cell division is going to be called mitosis. Mitosis is going to occur in 4 steps. You have your prophase, your metaphase, your anaphase, and your telophase. So let's talk about what happens in each of these steps, because each of them has a very specific function. Starting off with prophase. Now, before we start talking about the functions of prophase, I want to show you a difference in the top picture and the bottom picture. So here in the top picture, what you're looking at is a plant cell, because as you can see, it has a little cell wall that allows the cell to look like a little brick. And inside each of the bricks, you can see a little circle, fully intact. That's the nucleus. When the nucleus is fully intact like this is what the cell looks like when it's in interphase. So it's doing its day-to-day activity. When the cell is ready to move on to mitosis, the first thing it's going to do is it's going to enter prophase. And prophase, the main function of it is to start coiling up the DNA so it makes it easier to transport. And what's going to happen is we're going to release the DNA from the nucleus. So we're going to allow for the nucleolus and the nucleus to disappear. So under the microscope, what we would see is that that small little circle starts to become enlarged, almost like it's stretching out. And actually, here's a really good example. And inside of the nucleus, you start to see not only is the nucleus getting larger, but you also start to see these strings that are appearing. The strings are the DNA or the chromosomes that are coiling up very tightly. And the whole purpose of prophase is to coil them up tightly so they're more accessible and to get rid of the nucleolus and the nucleus, because you want your DNA to be freely accessible for the rest of the cell. Now, A little disclaimer, some books, including ours, will mention pro-metaphase. Pro-metaphase is sort of like an in-between phase between prophase and metaphase. And for me, I just talk about prophase. But in case you were wondering how do they divide it, you can see that the prophase is concentrated on the nucleolus. And then for the books that discuss pro-metaphase, that will be the part that they focus solely on the nucleus itself. But for us, we're just going to talk about prophase. So that would mean the DNA coils up, the nucleolus disappears, and the nucleus disappears. Once the nucleus is gone, we see that inside of the cell, now instead of a circle, we start seeing these little strings. That, of course, is your chromosome. But more importantly, we see that the chromosomes start to get organized down the middle of the cell. So it's almost like they're clustering together. When we see that, that then means that metaphase is occurring. So in prophase, we got rid of the nucleus, and in metaphase, we took all the chromosomes and we aligned them down what we call the equator, or the middle of the cell. This is, of course, done with the help of the mitotic spindle. So here's a little preview. We started off in interphase, where the cell was just doing its day-to-day activity. Here is an illustrated example, an animal model, and a plant model. So as you can see, the nucleus is fully intact. Nothing exciting is happening. Then when you enter prophase, we're going to see that the chromosomes become more visible because they're going to start condensing more. And we're also going to see swelling of the nuclear envelope. It's going to get larger, and that's because the nucleus and the nucleolus are starting to disappear. Once that's done, we enter into metaphase, which means that we take our chromosomes and we align them down the middle of the cell. And you can see these little darkened lines. That's our mitotic spindle. That's our protein complex that's moving the chromosomes around and organizing them down the middle. Now, the reason you want to have everything organized down the middle is because once that's done, you can move on to your next stage, which is anaphase. Anaphase says, let's split the DNA. And the reason we're doing that is because, as you may recall, you doubled all your DNA, so you have 92, and we're going to split them into batches of 46. So one batch of 46 is pulled to the left or to the top, and the other batch is pulled to the right or to the bottom. And as you can see, it's starting to separate. To me, it always looks like fingers that are being pulled apart. They were interlocked, and now we're separating them, and we're creating two separate batches. And that's what anaphase does. So anaphase will divide up the DNA by splitting the centromeres, what was holding them together, and pulling them into two separate batches. Once the DNA is back to its original number of 46, then in telophase, we have to go ahead and restore the nucleus and the nucleolus. So as you can see, it gets coiled up. And right now what we have is we have one cell that has two nuclei. So our last step is going to be this thing called cytokinesis. Cytokinesis means we're going to split the cytoplasm. It happens at the end part of telophase, and it's going to allow us to physically separate one cell into two daughter cells. And you can see that cytokinesis is about to occur because if we look at our slides, we start seeing this little line that runs in between the two nuclei. That is the cleavage that's starting to form, indicating that the cytokinesis, or the cytoplasm, is starting to split. In order for us to recall the steps of the cell cycle, so interphase, day-to-day, prophase, metaphase, anaphase, and telophase, those four are part of mitosis, I want you to think of the sentence, I passed my anatomy test. And that sentence, if we use the first letter of each word, is going to correlate with the first letter of each of the phases. So interphase, prophase, metaphase, anaphase, and telophase. Please keep in mind that the entire cell cycle will include all of them, but if you're only talking about mitosis, you start with prophase, because prophase, metaphase, anaphase, and telophase are the stages of mitosis that are needed to go from one cell to two daughter cells. So here we once again have an illustration of our anaphase, where we're going to be splitting the DNA to opposite ends of the pole. So you can see them moving into two separate batches. Anaphase is followed by telophase, which is us reforming the nucleus. So now we have two nuclei. We still have one cell, and that's because after telophase, we have to do cytokinesis. Cytokinesis is the division of the cytoplasm. As you can see, it happens at the end of telophase, and it's going to allow us to sever, to divide that one cell into two daughter cells. And these two daughter cells will then go ahead and start the cell cycle all over again in interphase. And they'll continue on. Interphase, prophase, metaphase, anaphase, telophase. So cytokinesis is the tail end. It is the division of the cytoplasm, and it's going to be towards the end of telophase. And it's shown, as I was mentioning to you before, by that little line that forms in between the nuclei. That little line is called the cleavage furrow in animals, and it's called the cell plate in plants. The reason plants get a different name is because, as you may recall, plants have a cell wall in addition to their cell membrane that has to be divided as we go from one cell to two daughter cells. Here is a nice little look at cytokinesis. You can see it happening here with our animal cells. You can see the cleavage furrow that starts to form, and here it is from another angle. So this will allow us to see that cytokinesis is occurring. Plant cells do the same thing. They have the little line. You can see it right here. And then once we see that line, instead of calling it cleavage furrow, we call it cell plate because it's going to allow the division of the cytokinesis. And of course, for the two daughter cells to have a cell membrane and a cell wall, just like the original cell that they started with. So the cell cycle is interphase, prophase, metaphase, anaphase, and telophase. And at the end of doing mitosis, you walk away with two daughter cells that are identical in DNA as the original cell that you started with. In case you were wondering, how does the cell know when to move from one step to the next? The reason the cell knows how to go from like interphase to prophase to metaphase to anaphase to telophase is because there are proteins called cyclins, and they are able to fluctuate the cell from one stage to the next. So these cyclins are the guide that move them from one step to the next. Along the way, they will come across what we call checkpoints, and checkpoints will be areas in the cell where the DNA is scanned and observed to make sure that they are ready to move to the next stage, and also that there are no mutations. If there is a mistake detected in the checkpoint, then one thing that the cell can do is it can induce apoptosis, programmed cell death. So that way that no mistakes are moved on and that the cell can start fresh again. An example of a checkpoint is the p53 gene. The p53 gene is well studied because scientists like to study how it can induce apoptosis. And if someone has a mutation in their p53 gene or the gene doesn't work properly, it puts them at a higher risk category of having mutations. And the more mutations you have, the more likely you are for cancer to develop in the body. So as you can imagine, your body wants to protect itself as best as possible. So it wants to actively regulate the checkpoints and make sure that no mutations pass on by doing apoptosis when a mistake is detected. So here you go, we can see an example of our different cell cycle checkpoints. So we have our interphase, and as you can see here by the little stop signs, there are different checkpoints where the DNA is checked. Is it damaged? Did it replicate properly? And then even when you go into your cell division, when you go into your metaphase, for our example, and you do your prophase, your metaphase, your anaphase, your telophase, the checkpoints will go ahead and run to see is the protein properly splitting, is the spindle properly splitting the DNA, is the nucleus properly forming. This is a fantastic way to, of course, control the quality of the work. And like I mentioned to you before, if this doesn't control, it does lead to mutations, and mutations make it more prone for cancer to develop. In fact, a cancer cell, by definition, doesn't follow the rules of the cell cycle and will just replicate uncontrollably and is often able to avoid apoptosis. If you would like to learn more about cancer, I highly encourage you to read the end part of the chapter because it will talk about all the different symptoms and the different types of cancers that are out there. And of course, if you have any questions, comments, or concerns, please do not hesitate to reach out and I'll be more than happy to assist. All right, everyone, let's call it a day and we shall talk again soon. Bye.